Hello, I am Dr. P.R. Padma, Professor of Biochemistry at the Avinashlingam Institute for Home Science and Higher Education for Women, Coimbatore. In this session, I shall be dealing with the introduction and the applications of genetically modified organisms. The organisms that have been genetically modified are called genetically modified organisms, or more popularly GMOs. This module gives an overview of the enzymes and the methods involved in the genetic modification, as well as outlines the major applications of genetic modification of organisms. After completing this module, you will be able to know the definition and the concepts involved in transferring genes to organisms, understand the enzymes and the methods involved in gene transfer, and learn about the major applications of genetically modified organisms. This module is part of the Food Biotechnology paper. It is designed to give an overview of the definition and the concepts of gene manipulation, its insertion into a vector, the nuances of the creation of genetically modified organisms, and their applications. From time immemorial, man has taken several efforts to improve the properties and the traits of plants by selective breeding and crossbreeding. By doing so, he has been practicing genetic modification without actually knowing it. However, in these efforts, the modifications were taking place within the same species of the plant. Today, with the advent of DNA manipulating tools, it is possible for us to make specific modifications in the genetic sequences and carry or insert desired sequences of genes responsible for specific traits into any organism crossing species barriers. This process results in the creation of genetically modified organisms, or GMOs. GMOs are generally plants or animals that contain alterations in their genetic material and hence genetic characteristics which does not occur by natural means like pollination or mating. The genetic alteration is caused by artificial means, by purposely inserting or deleting specific sequences of DNA in the genome of the organism. Such genetic modification is carried out to achieve some desired effect like better growth characteristics or production of a desired product by the organisms, and so on. Genetic modification is also carried out on microbes very commonly to achieve desired traits. Let us now see how GMOs are made. Genetic modification typically involves the insertion of a new sequence, which is the gene of interest, or GOI popularly, into the genome of the recipient organism. This insertion cannot be done directly and requires a carrier molecule to take the gene across. Such carrier molecules are referred to as vectors, which are also DNA sequences. Additionally, multiple copies of the gene is required, which is also achieved by amplification after insertion into a vector. Let us now consider how this is made possible. Essentially, to insert a new piece of DNA into another piece of DNA, we need to carry out some specific steps. Isolate the DNA to be inserted, which is otherwise called the gene of interest or the insert sequence. After this, we cut or trim the DNA to be inserted. Then, we cut open the recipient DNA and put the DNA to be inserted in proper place in the recipient DNA. Finally, we seal the ends together to make a single piece of DNA with the inserted sequence in position. Nature has provided all the material needed for such modifications in various organisms. Scientists have identified and are exploiting these materials to carry out very precise cutting, insertion, and sealing of the sequences. Let us start understanding each of these steps, starting with the isolation of DNA to be transferred, or the insert DNA. The insert DNA is the gene that one wishes to transfer from one organism to another for the creation of GMOs. This DNA is identified and isolated from the cells in which a desired trait has been identified and needs to be expressed in a new location or a new organism. In-depth research into the biochemistry involved in various metabolic as well as disease processes has enabled the identification of the proteins that are major players in these processes. Once the protein is characterized, the gene that codes for the protein is identified. Many a times, the gene involved in such processes can also be directly identified by various techniques. Once the gene is identified, it can be considered as a gene of interest for the preparation of recombinant DNA. For this, the first step is to isolate the DNA and prepare it in a pure form. Several methods and ready-to-use kits are available to isolate and purify the DNA sequence of interest. Let us see the basic steps involved in the process. First, we disrupt the plasma membrane of the cells containing the DNA of interest using physical methods like mechanical shearing or chemical methods like the use of detergents or biological methods like using enzymes. Once the cells are lysed, their contents flow out, and in the next step, we degrade the proteins present in the released material. This is usually accomplished by the use of phenol, which precipitates protein, or proteases, which are enzymes that can specifically cut down protein molecules. The next step is to degrade the RNA present in the released material using RNase, which is an enzyme that specifically cleaves RNA. The last step is purifying the DNA that is left using methods like column chromatography or affinity binding to specific support. This procedure gives the DNA in pure form. The DNA thus isolated is cut using enzymes to prepare fragments of varying lengths. From the fragments, the one containing the desired sequence can be separated by electrophoresis and probing. This fragment is then trimmed using restriction endonucleases. Now, the insert DNA or the gene of interest is ready for use in the recombinant DNA preparation. When isolated from the cells, the DNA will be very low in concentration. This concentration is usually increased in an enzyme catalyzed process outside the cells. This process is called amplification of DNA, which is discussed later in this module. Now, let us consider the vector DNA. The second piece of DNA needed for cloning of the recombinant DNA to be produced is the vector DNA. This is the DNA sequence that carries the gene of interest or the insert DNA into the recipient cell and puts it in place. There are a large number of vectors that are being successfully used for such genetic engineering processes. These vectors are from varied sources, right from very small viruses or phages and prokaryotic bacteria to pieces of human DNA too. The choice of the vector depends on the size of the insert and various other factors. Bacterial vectors can carry DNA of small to moderate size, while the artificial vectors constructed from bacterial, yeast or human DNA can carry insert DNAs of large sizes. Vectors can either be circular or linear in nature. An ideal vector should have certain specific characteristics. We will now list the most important ones. A good vector should be capable of replicating and expressing in the host cell or the recipient cell. It should have convenient restriction enzyme sites for inserting the DNA of interest. The vector should have a selectable marker to indicate which host cells received the recombinant DNA molecule, and the vector should be small and easy to isolate and handle and easy to construct. The most common classes of vectors include the bacterial plasmids and phage DNA. Plasmids are circular DNA molecules that are present in the bacterial cells. These molecules do not carry genetic information of the bacteria but are important in conferring certain special properties like antibiotic resistance to the bacterial cells. The plasmids are ideally suited for cloning experiments because of two important characteristics. Firstly, they can replicate within the bacterial cell independent of cell division. And secondly, they contain sequences with multiple cloning sites, which are characterized by the presence of many restriction enzyme recognition sites. This enables them to be cut by a restriction endonuclease that best suits the DNA to be inserted. A representative figure of two commonly used plasmids is now displayed on your screen. These are PBr322 and PUC18. In both these plasmids, several restriction enzyme sites are present in sequences called polylinker sites. PBr322 and PUC18 are commonly used vectors for the insertion of DNA molecules up to 20 kilobases long. As shown in these figures, both the plasmids contain ampicillin resistant sequences, which enable the selection of the successfully transformed cells easily by plating onto ampicillin containing medium. In addition to this, PUC18 plasmid also contains the LAC-Z gene sequence, which can convert chromogenic substrates like IPTG and XGAL to a blue colored derivative. This makes the entire colony of successful transformant cells appear blue in color, which can be easily identified and collected. Another commonly used vector is the bacteriophage lambda DNA. Lambda DNA is a linear DNA. It contains within its structure a small sequence about 15 kilobases long that is not required for its insertion into the bacterial cell or for its replication within the bacterial cell or for the reassembly of the phage particles. This piece of DNA can be excised from the lambda DNA and a new piece of DNA, that is your gene of interest, up to around 25 kilobase size can be inserted in its place. Thus, the lambda DNA is also an ideal vector for cloning experiments. This is a schematic representation of the lambda DNA. The site that is marked as recombination is used for the insertion of foreign DNA, and these sequences are used for the construction of recombinant DNA. Plasmids and phage DNA are naturally occurring DNA vectors. Since they can carry insert DNAs with limited size, the need for vectors that can carry larger inserts was intense, as genes often can be much longer. Hence, several synthetic vectors have been constructed and used successfully. Combinations of plasmids and phage DNA have been developed into good vectors, and these are termed as cosmids. Similarly, as we mentioned earlier, artificial chromosomes have been constructed from bacterial DNA to form bacterial artificial chromosome or BAC, yeast DNA for yeast artificial chromosome or YAC, and even human DNA for human artificial chromosome or HAC. These vectors are capable of carrying inserts that are very large in size. Another major vector that is widely used in plant transformation is the TI plasmid. This plasmid is present in a bacterium called Agrobacterium tumefaciens. This is a soil microorganism that induces crown gall, which is a type of tumor in plants. The tumor is induced by inserting and integrating the TI or tumor inducing plasmid into the plant cells. The TI plasmid also has been modified to carry large pieces of DNA inserts. Since it contains the natural ability to insert and integrate itself into the plant cells, the transformation of cells becomes much easier using the recombinant TI plasmid carrying Agrobacterium tumefaciens. As shown in this figure, the tDNA region of the TI plasmid is the sequence that is transferred to the plant cell infected by the bacterium. Therefore, this is the region that is used to insert the gene to be transferred into the plant cell. The TI plasmid is a preferred vector for several plant transformations. Thus, the choice of the vector DNA depends on several factors, including the size of the insert and the recipient cell where the inserted DNA is to be expressed. Now we shall move to the amplification of DNA. The number of copies of any given piece of DNA can be amplified in vitro using a technique called polymerase chain reaction or PCR. In this process, the DNA to be amplified is taken into a reaction mixture containing the monomers of DNA, that is deoxyribonucleotides of adenine, guanine, thymine and cytosine, a buffer to provide ideal conditions for the reaction to occur, and an enzyme named DNA polymerase. The DNA polymerase used in PCR is highly thermostable and can withstand a wide range of temperature without loss in catalytic activity. This is because the enzyme is isolated from thermophilic bacteria or heat-loving bacteria which live in high temperatures like the hot springs. The most widely used enzyme in PCR reactions is Taq polymerase that is isolated from a thermophilic bacterium named Thermus aquaticus. A typical PCR reaction mixture contains the following ingredients. 1. The template DNA or the DNA to be amplified. 2. Thermostable DNA polymerase like Taq polymerase. 3. 2 DNA primers to initiate the polymerase action, which are oligonucleotides that are complementary to the 3' ends of the sense and the antisense strands of DNA, termed as forward and reverse primers respectively. 4. The monomers or building blocks for the building of new DNA strands or the deoxyribonucleotide phosphates. 5. Cofactors required for the enzyme action, namely magnesium and potassium ions. And 6. A buffer solution to provide the medium for the polymerase action. This reaction mixture is taken in thin-walled PCR tubes and placed in a PCR thermocycler, which is shown on your screen now. The thermocycler contains a block into which the tubes are placed. This block can be heated and cooled to desired temperatures, which can be controlled by microprocessors. Since the PCR tubes are thin-walled, the temperature is quickly conducted to the reaction mixture. In a typical PCR reaction, as shown in this figure, a typical series of temperature settings are used. Heat to 94 degrees centigrade to melt the DNA or separate the DNA strands. Cool the mixture to enable the binding of the primers to the DNA strands, which is called annealing, and heat to 72 degrees centigrade for the action of Taq DNA polymerase to take place, which is a step called elongation. These 3 steps are repeated 30 to 40 cycles when multiple copies of the DNA are generated. This process is called amplification. Finally, the reaction is stopped and the DNA is collected either by precipitation from the reaction mixture or by passing through a column and eluting with a suitable agent. With this step, the DNA to be inserted is available in sufficient number of copies and can therefore be used for trimming using restriction enzymes. The vector DNA is cut open by restriction endonucleases. The same enzymes are usually used to trim the gene of interest also, so that the cut ends are compatible with each other. When the 2 pieces of DNA, that is the insert and the vector, are mixed together, they generally align themselves automatically due to complementary base pairing. Then, an enzyme named DNA ligase is used to seal the ends to form a continuous piece of DNA. Let us now consider these steps in detail along with the material needed. Let us start with restriction endonucleases. Nucleases are enzymes that can cleave or cut nucleic acids. Endonucleases cut the DNA at sites within the sequence, unlike exonucleases that cleave the molecules from the ends. Restriction endonucleases have specific recognition sites and are very specific in their action. There are three classes of restriction endonucleases, namely type 1, type 2 and type 3 endonucleases. Among the three classes, the type 2 endonucleases are more commonly used in genetic engineering and hence are more commonly used in the preparation of GMOs also. These enzymes have palindromic recognition sites that are 4 to 6 nucleotides long. Palindromic sequences are those that are read as the same sequence when the sequence on the forward and the reverse strands of DNA are compared. For instance, the recognition site for the restriction enzyme EcoR1 is GAATTC. In the opposite strand, the complementary sequence is CTTAAG, which, when read in the reverse or anti-parallel direction, is the same as the first. Thus, it is a palindromic recognition site. The restriction endonucleases can cut DNA in two ways. One type of cut is straight and the other is staggered. The straight cut results in blunt ends in the resultant DNA pieces. The staggered cut results in small overhangs of sequences which are complementary to each other and can readily pair with each other. Therefore, they are called sticky ends. When two different pieces of DNA are cut with the same restriction enzyme, the ends of these pieces are complementary to each other and can base pair to one another. Enzymes that can form sticky ends are preferred over those that cause blunt ends in genetic engineering. For example, the action of EcoR1 is represented in this figure. As you can see, the enzyme produces sticky ends. When two pieces of DNA, that is the insert and the vector, are cut with the same restriction endonuclease, they will contain complementary overhangs. When they are mixed together under physiological conditions, they will readily pair at these complementary overhangs and will be ready for sealing with another enzyme. Therefore, such overhangs are called sticky ends. Because of the ease in pairing, sticky ends are preferred over blunt ends for genetic engineering attempts. However, sometimes a recognition site for an enzyme causing sticky ends may not be available in the vector and or the insert sequence. In such cases, restriction enzymes causing straight cuts resulting in blunt ends may be used. Thus, the vector and the insert are cut using restriction endonucleases and mixed together when the insert DNA or the gene of interest gets positioned inside the vector sequence. Now, the ends have to be sealed together to get a continuous sequence. This is called ligating the insert DNA with the vector DNA. That is, the ends of the vector and the insert DNA are sealed together in an enzyme catalyzed process called ligation. This sealing is mediated by an enzyme called DNA ligase. The process is represented in this figure. DNA ligase catalyzes the formation of phosphodiester bonds between the ends of DNA strands that need to be sealed together. Several sources of DNA ligases have been tried in the production of recombinant DNA, including bacterial and viral sources. The most widely used enzyme is the T4 DNA ligase isolated from the bacteriophage T4. This enzyme is capable of sealing both sticky ends and blunt ends of DNA. T4 DNA ligase acts optimally at 16 degrees centigrade. When the action of DNA ligase is complete, the DNA is sealed without any gap, resulting in a new piece of DNA with the vector containing the gene of interest as part of its sequence. This is the recombinant DNA, which is now ready for transformation into recipient cells. The entire process of formation of a representative rDNA is represented in the figure displayed now on your screen. The recipient cells of the recombinant DNA or rDNA can be microbial, plant or animal cells depending on the application desired. When the rDNA is inserted and expressed in plant or animal cells, the resultant organisms are transgenic plants and transgenic animals respectively. They are the genetically modified organisms or GMOs. The newly inserted sequence that leads to the formation of a transgenic organism is called a transgene. The transfer of a transgene into plants and animals can be mediated by several physical, chemical and biological means which are discussed in detail in the next module, that is module 32 of Food Biotechnology paper. Now let us consider the applications of genetically modified organisms. GMOs have several applications that include conferring useful traits, production of valuable products and pharmaceuticals, as well as in research to understand complex biological processes. Some of the major applications include the production of insect and pest resistant crop plants, herbicide resistant crop plants, drought resistant crop plants, GM plants that contain improved amounts of nutrients like the golden rice that contains the transgenes to synthesize beta carotene, GM plants that can produce pharmaceuticals and biofuel, GM traits to delay the ripening as well as to reduce spoilage of food, GM animals that can produce more milk and more meat, GM salmon that can grow fast as well as much longer. The list goes on and on. Another major application of genetic modification also includes the conservation of diminishing species and exotic species of plants and animals. The major applications of GMOs in food technology are discussed in detail in modules 33 and 34 of this same paper, that is the Food Biotechnology paper under Food Science and Nutrition, which will give you a complete understanding of how important GMOs are for food technology and food industry. Thus, in this module, we have considered in detail the various steps involved in the production of a transgene which is used for the production of genetically modified organisms. Each of these steps are crucial and have to be carried out with meticulous care. Many successful constructs of the transgenes have been made and transferred into plants and animals for the production of GMOs. These find vital applications in various fields, including the food industry. The nature of these organisms and their applications in food technology are discussed in the subsequent modules. With this, wishing you all a happy learning experience, this is Dr. Padma signing off. Thank you.